Okay, hello everyone. My name is Farshad, and today I'm going to talk about some basic results on range evolution based on some PD models that have already been developed and we have extended a little bit. And it's basically based on two hypotheses on range evolutions that I will talk about, which are trying to address these fundamental questions in ecology, that we need to know that why are species range limits where they are, because we often see that range limits are set in some places. We don't see any obvious physical barriers or habitat discontinuities, and knowing the fact that the species are able to adapt to dramatically different environments, then this raises a question that what factors are preventing adaptation and allowing the further range expansion of this species. There have been many hypotheses on that. I'm going to talk about two of them. The first one is due to Hallin and Mayer. It's called genetic swamping hypothesis. In this cartoon, imagine this green gradient is showing you the gradient in a fitness, in the optimum value of a fitness-related trait imposed by the environment. And then we have a population there. And the individuals in the population can disperse randomly, but since the population is more crowded at the core, that random movement which is going to create some random gene flow is going to be asymmetric from the core to the periphery of the population, because there are more individuals which are moving to the periphery compared with the individuals that are moving from the edge to the periphery. And since individuals at the core are pre-adapted to the locations that they were, once they move to the edge, they are not adapted there. And that creates a migration load, which can destabilize the migration selection balance, and then can swamp the adaptation at the edge and then prevents range expansion. The other hypothesis is due to Darwin himself, and it's just saying that interspecific competition can be a major factor in setting range limits between two competing species. So there have been a sequence of models. The highly influential one starts with Kirkpatrick and Barton model, which they try to model the joint evolution of population density and mean value of the trait by assuming the trait variance is going to be constant, which might be an unrealistic assumption in a one-dimensional space. And then they conclude that if the environment is sufficiently steep, then genetic swamping is going to cause range limits. Okay. But a few years after, Professor Barton himself relaxed this assumption of genetic swamping and then concluded that no, genetic swamping, the assumption of constant variance, and then he concluded that genetic swamping cannot cause range expansion no matter how steep the environment is. But the result is hidden in a book chapter so that nobody can see it. So, another major contribution comes from Case and Taper. In that model, they extend Kirkpatrick-Barton model to a community of the model which are interacting competitively. And then they show that interspecific competition can interact with gene flow and can set range limit in much shallower and more realistic environmental gradient. So what we have done is that, but the Case and Taper model is still based on constant trait variance assumption. What we did, we first relaxed that assumption of Case and Taper. We allowed for the joint evolution of trait variance with trait mean and trait and population density. We observed that genetic swamping hypothesis could not limit the range of the species, but interspecific competition could, even in the case that we had evolving trait variants. But it happens that when the environment is linear, the range limits set by interspecific competition cannot be stable. The stronger species is going to push the weaker one toward the edge of the habitat. There are some stabilizers that have been discussed in the literature. We identified certain environmental nonlinearities, which we call them knees and wiggles, for a reason that you will see. And these nonlinearities can strongly stabilize the range limits, and this stability is going to be robust against pretty moderate, like strong climatic changes. And then we relax another assumption, which is quite typical in this sort of range evolution models, which we usually assume that the movement of the individuals, the dispersal, is random, which is often not the case. And genetic swamping hypothesis relies strongly on this assumption. So we relax that, we allow for a sort of like optimal dispersal, which is known as matching habitat choice. Individuals move to the place where the optimum is going to best suit their phenotype, to maximize their phenotype environment match. Okay. Then we concluded that this mode of adaptation, this mode of dispersal is likely to evolve only in environments which are very steep. And if it does occur, it has substantial impacts. It can create adaptation in single generation time scale. It can substantially reduce trait variance. It can create adaptive gene flow instead of maladaptive gene flow. Means that it is going to rule out the possibility of genetic swamping hypothesis entirely. And it has some like side effects like promoting phenotypic segregation, assortative mating, sympactic speciation, and these sort of things. All right. These were the summary of the results that I want to talk about. And for the rest of my talk, I'm going to show you the details as much as I can, but I will have to stop at the time that Daniel tells me. Okay. Yeah, the model is developed at the macroscopic phenotypic level. So this is the basic equation underlying the development of the model. In the evolutionary game theory, we usually call this replicator dynamics. So let's say that I'm denoting the phenotype by P is a random variable, and I don't know, you can see this. And then let's say that the distribution of P at a location x times t is given by this density function phi for the ith population in the community. Okay? So phi is just the probability density function. Let's say that the density of the population at location x and t is denoted by n. Then n times phi is going to be the density of the individuals with phenotype P. Then the change in that density over a short time interval tau is given by these three factors. A diffusive movement, this is the divergence, this is the gradient, this is the divergence, the diffusion rate. So the change is coming from three factors, like random movements, diffusive movements, intrinsic growth of the population denoted by G, and then mutational changes. Okay. And the ingredients, if I want to just briefly show you, are pretty classic. For the fitness function or the growth rate, we have just like logistic growth. We can have allele effects also. And then we have like carrying capacity, maximum growth rate, and then competition kernel. This term models the effects of stabilizing selection. Q is the value of the optimum trait, is penalizing the phenotypes that are far away from the optimum. S is the strength of stabilizing selection. Competition comes from the MacArthur-Levin's overlap between these resource utilization or phenotype utilization curves. There is a parameter K that allows for the asymmetric competition and these sort of things. Then for the mutation, we assume that the mutation rate from a phenotype P prime to P is going to be dependent only on the difference between P and P prime, and the distribution on this difference is going to be normal with zero mean and constant variance. Okay, this is also like a classic model of mutation that we found in the literature. Then, if we substitute these ingredients in the Sorry, where did I go? If you substitute the ingredients in this basic equation, then you integrate to find the first moment and second moment. You use this assumption that phi, the distribution of the phenotypes, is normal for every x and every t. This is a strong assumption. But this assumption, if we leverage it, it allows for exact moment closure. And then we can find the moments, we eventually find the equations of the model, the joint evolution of population density, trait mean Q, and trait variance V. These are the equation. And there is a non-intuitive term, gradient of natural log of the population density, which appears there, just creates some pain for us for analysis, because it's going to be quite difficult. And then there are some like on nonlinear mappings, G, H, and W. They are going to look like this. Then they have some other nonlinear terms there. I'm just showing you this that to convince you that the equations are large. So we have no choice but to analyze them numerically. But if you want to do that, you need to pick right values for the parameters of the model to be biologically reasonable. And if you want to do that, first you need to choose the units for those parameters. And it's quite challenging for this sort of models even to find the right units for the parameters to give sufficient generality to the model. So what we did, let's say that we pick a representative population, we calculate the mean generation time, we set it as a unit of time. And then with that, we calculate the root mean square of the dispersal distance, and we set that as the unit of space. Once we have that, we measure the carrying capacity of the environment, and we set the population abundance unit such that the carrying capacity is. And after that, we measure the trait values, we calculate the standard deviation of the trait values, and we set that standard deviation as a unit of measuring the trait values. And with these parameters, we could search in the literature. We spend a lot of time. We could find reasonable values for every parameters in the model. The difficult one is going to be the steepness of the gradient in the trait optimum, which is hard to estimate. And we couldn't find much of results. For that, you need to measure the trait values, find the standard deviation. You need to measure the optimum trait value. You need to measure the dispersal distance and also generation time. All these are involved because of the units that I have chosen. And we just found some rough estimates suggested by Case and Taper based on their units. And then we found like 6,000 different species for which we had dispersal rate and also generation time. And then we tried to scale the suggestion of Case and Taper. And we found that the optimum, the steepness of the gradient value is going to be between 0 and 2 mostly. If it is larger than 2, like 10, it's going to be like something like a physical barrier or something like that. Now, if I want to show you some typical range evolution that we see for a single species, if we initialize the population by this orange curve, it's going to establish itself to the carrying capacity and then expand in the form of a traveling wave. And if you have a trait optimum, which is shown by this black line, and then the population is also going to adapt to that trait optimum gradually. If you measure the traveling speed of the range like expansion and also the magnitude, the maximum population density, you plot them for different values, for different steepness of the gradient, you will see these two curves. The green curve is coming from Kirkpatrick-Barton model based on the constant trait variance assumption. And the purple one is what we get when we relax that assumption and the trait variance can evolve. So in Kirkpatrick-Barton, you see that there is a critical gradient at which the wave speed goes to 0 while population density is non-zero. That is the bifurcation point where genetic swamping is going to pin your ranges. But we don't see such a thing here. We just see that when the trait variance can evolve, the wave expansion speed goes to 0, but at the same time, population density also goes to 0. So there is a critical gradient at which the population is going to go extinct. But we don't see any range limits caused by genetics. We could calculate, even though the equations are complicated, but this by a little bit of algebra, we can calculate this critical gradient and find this closed form. And what this is telling you is something that is just not, is probably counterintuitive. It tells you that if dispersal is lower, range expansion speed is going to be higher. And the chance of survival is going to be higher. And the reason is that higher dispersal means that higher maladaptive gene flow, and that is detrimental. Okay? But this is strongly based on the assumption that dispersal is random. Once you make it not random to be optimal, all these stories are gone. So, but based on this fact, there have been studies in the literature that they try to say that in steep gradients, species is going to evolve lower dispersal in order to survive. But that comes because of the assumption that they have that the gene flow is random. And for two competing species, if you initialize them allopatrically, once they meet each other, competition is going to cause their density go down. Once the density goes down, they get asymmetric gene flow because the density at their periphery is now lower than the density at the core. That creates some asymmetric gene flow. That asymmetric gene flow is maladaptive, is going to actually create and intensify some sort of like character displacement, and this is going to result in more decrease in the population density. More decrease in population density means that more asymmetric gene flow. This is going to be a reinforcing loop that is going to happen, and that eventually destabilizes the competition selection balance, and at some point the population density goes to 0, and this way we get the range limit established there. And when everything is settled at the equilibrium, we can see that the two species are showing character displacement over their region of sympatry. But as I said, when the environment is linear, these range limits formed by the competition is not going to be stable. The stronger one is going to push the weaker one to the habitat boundary. That can result in the exclusion of the weaker one if the difference is too large. If the difference, for example, I'm showing just the different growth rates, maximum growth rate. But if the difference is not very large, we have a chance that the weaker species is going to survive marginally near the habitat. And the reason is that once this species is contracted toward the edge of the habitat, it loses its core. So it suffers less from the core to edge maladaptive gene flow. And that advantage is going to compensate for the weakness that is the species. And to stabilize these range limits, there have been some ideas. We identified these environmental nonlinearities at which the steepness of the optimum gradient changes sharply. We call them environmental knee or even environmental wiggle, which is just like two changes like back and forth. And these can be prevalent in the nature, especially this one can be like ectones. What we see in ectones is like sharp changes in the environment. And the two species, when we initialize them, initially here, they are going to meet each other. The interface is going to move because the orange one is the stronger one. On the other side, the green one doesn't see any competitor, can establish itself entirely to the available habitat. But once the interface between the two species reaches this environmental knee, it's going to get stuck there, stabilized as an equilibrium, which is going to be an asymptotically stable equilibrium. And why that happens, there has to be something happening in this knee that gives some advantage to the weaker one. And that is exactly coming from the maladaptive gene flow, because the weaker one has already had the chance to establish itself to the shallower environmental gradient. As a result, it is suffering from less of maladaptive core to edge gene flow. And once the interface reaches there, the stronger species is still suffering from a lot of maladaptive gene flow because of the steepness of the gradient there. And that way the range gets stabilized. And the stability is pretty strong. When you change the environment, I just shifted the trait optimum by like magnitude of 4 over a duration of like 10 generation times. And then the two species try to adapt themselves to the new environment. The stronger one has a little bit of advantage. It shifts the interface a little bit to the right. But once the two species get adapted, the weaker one can catch up and then push the interface back to the equilibrium. So the message for ecology can be the fact that environmental needs and like vehicles can be the regions that are in which the range limits are less sensitive to things like climate change. Just to show you what you missed, because I don't have any time, we included matching habitat choice, in which individuals can choose their habitat based on their phenotype. Colors here, moving to their matching habitat. It has been tested in nature that for camouflage-based background matching in grasshoppers, for example, or for like temperature-dependent performance for ciliates. And what we can include that in our model by adding an advection term. That advection term comes from the idea of drift diffusion equations. The movement is going to be created by a force induced by a scalar potential energy, which is based on the perceived mismatch of the individuals and the gradient. And that can change some equations in different ways, can give different like things. Oh, you said okay. Oh, these are the things that you missed. Sorry.